Well, thank you very much for this very kind introduction. I was standing in the corner blushing, really. It's an honor. It's an honor and a pleasure to receive the award, not only for the award, but also that you get the chance to give a talk. It's basically carte blanche to talk about anything you like. Are you really sure about this? It will be okay. It will be sort of a fairly normal scientific talk. And also, Roland was very good at keeping the titles, which I know you do here in Germany. I'm not sure I will be able to do that because the normal Swedish way is addressing people by first names. It's not out of respect, but it's just different culture. So I'm just giving you a heads up on that one. As you said, I work at RISE mainly. I also work at Chalmers in the same department as Roland, so it's a mixture. RISE is like Fraunhofer, if you don't know. So our main purpose there is to help industry through research and through applying research. And much of this talk will actually be on applied research also. But as we saw yesterday, the theme this year for the award presentation is Save the World. Julie gave a very nice start of yesterday, so I will continue on that theme as well. So today's topics: how to save the planet and the secret of healthy aging, and of course virology. This may sound a little presumptuous, but I will actually give some examples where virology can contribute to make a difference, really. But then this is a chance to do something a little bit unusual. So I thought I would address the young scientists especially. It will not be very much, but maybe a little here and there. Because you're really the future of the society and the future of the field. And I remember when I was at these first lectures or conferences, there was also always some guy an older guy who I didn't have a clue who he was. And they talked very nicely about him, and they gave a presentation, and usually it was pretty good, but I didn't know who it was. And this is the same thing here. And you're saying that 32 years in the board. By God, what is he doing? Why was he doing that? So I mean, I'd like to say a few words about myself, so you know at least the background and why I talk about things I talk about. So I started with a master's in engineering physics. Then I spent a year as a medical technician at the hospital, and realized that medicine also quite interesting. And then I studied some more chemistry and biochemistry to understand more on how we work, and also some very basic physiology and anatomy and this. So, and this means, and also I say I was never going to have a PhD, but yet here I am. But that means I'm more broad than deep. So I have a general knowledge of a lot of areas. I do neurology, of course, but otherwise this is sort of a killer for an academic career. So if you want to be a professor quickly, the young ones, don't do this. I mean, you should know your field. Or for somebody, this works as well, so you don't know. And outside work, it's not by purpose, but I sort of do the opposite what I do at work. I try to stay outdoors instead of indoors. I can speculate freely, which I never have a chance to do when I write a paper. And then I like to travel. I like to create photo and video. You probably have seen this here. And I'm recording myself here. So that will be the next project to see if I can edit that to something. And my latest project is also to make a YouTube channel about bike touring. It's a bit nerdy, but it's fun. And I'm not going to tell you the channel now, because then you just watch the videos. Okay, without more about background, let's go into the science part of this. So I will start with fluids, then I will move over to gels, and then also application of some more solid nature. So I will not be talking about the fluid, the slime you see here. I will talk more about what you eat. And that brings us to the first Save the World thing, or a societal challenge. I mean, we are getting older, and it's actually a lot older now than if you just look back 50 or 100 years ago. I mean, in Sweden, 20% are older than 65, and Japan is the extreme at 30%. I think Germany is probably close to 20 as well. And this is a good thing, right? I mean, we live to a higher age. But it also means that some of the problems associated with age has to be dealt with, and one of these are frailty. I'm sure a lot of you have some older relative who's getting thinner and thinner, and that's not good. And it's usually because they don't eat enough. And the reason they don't eat can be that they don't exercise, and then they eat even more, and then they do exercise less, and yeah, it's sort of a vicious circle, and you end up with losing muscles, and then usually it ends up with a fall accident, and you go to hospital. Unfortunately, it's not noticed before. So the older seniors need to eat more, and then you can answer, so what is really stopping them? And the major thing is what we have heard in some other talks in this conference. A lot of elderly have swallowing disorders. There are other reasons as well. It could be trauma or other states. But the common thing is age. And I mean, the body starts to sort of degenerate when you're like 25 or 30, and then it's just downhill. Sorry for the dull start here, but it's not so bad, because I'll show you that we also compensate for this. But that means when you swallow something, a lot of things has to happen at the same time. You have to keep your breath, you have to close the opening to the airways so the food can pass. This is a lot of signaling, a lot of muscles, and it gets slower and slower with time. Eventually, the older body doesn't have sort of strength and time to do this. And what you can see here is video fluoroscopy. The top one, the food goes the wrong way, so it goes into the airways, or partly into the airways, which is very uncomfortable. So the solution to this is, now comes the role, is thickening. So you add thickeners, and that means you have a slower flow, so the old body has time to adjust. It's very simple. There is one more reason, I'll come back to that. And what you see, all the products here, that you get a can with thickeners, you add it to your coffee or to your beer or to your whiskey, and that sounds pretty disgusting for us who don't need it. If you try, it's actually not so bad, because you just have to thicken it a little bit. And also, if you talk to the ones about who had this problem, they basically say it saved my life. Because if you can't drink, that's quite horrible. This is a compilation of a lot of different studies, and you see aspiration, like fluids in your airways goes down, and the safe swallowing goes up. So thicker is better until you start to have fragmentation and stickiness, and so there is a limit. But also elasticity is good, which we have studied. So that's the other thing. You want the bolus to keep together when you swallow. And there was a nice example of a simulation yesterday. And because if it keeps together, it can pass over the airways, even if it's not totally closed. So elasticity actually helps. We tried this with some model fluids. We had a Newtonian, we had a Booger fluid, which has constant viscosity, but you add a little polymer, so it's elastic. You can see it's not perfectly constant viscosity, but almost. And then we had a shear thinning. And for some reason, everybody measures at 50 reciprocal seconds in the swallowing field. Most foods are not really elastic. There are some examples, as in natto in Japan. We have ropey milk in the north of Sweden. But I mean, we don't really appreciate sliminess in our food. In Japan, there is actually a word for pleasant sliminess, which is like neba. It's a culture-dependent thing as well. So we worked with a hospital, and they had some patients, so which they had sort of evaluated for swallowing disorders. They did video fluoroscopy, and then they also tested these model fluids. They were based on maltodextrin and xanthan, so it's fairly simple fluid. And you can see that when they scored how easy it was to swallow, more of them scored the one which was elastic, which is the Booger and the shear thinning, higher than the Newtonian one. And then we also measure this pharyngeal transit time, how long time it takes for the bolus to pass. And this is the healthy one. You can see all of them have much more. You can't really say if one is better than the other. But if you count the number of the ones that with the fastest pharyngeal transit time for the different ones, again, the elastic ones are better. It's difficult to conclude that elasticity is good for everybody, but this and other studies actually indicate that elasticity is good. And you have to balance it to elasticity or sliminess, of course. But then we realized we had 12 patients here, and that's what we could get. And these are quite frail patients, and you don't want to bother them too much. So then we started building a model of the throat instead, because then we could do a lot of the experiments in the lab, and then we could try it on patients later, just when we had to do it. There was already a Cambridge throat, so we called it the Gothenburg throat, of course. And it works in the way that the mouth is simulated by pistons, so you can inject a bolus of fixed volume and fixed speed. And then you can observe it when it passes through the pharynx, the upper throat, and down to the esophagus, and that's where the stomach starts. And the dimensions of the pharynx are the ones that you have when the bolus passes. Otherwise, it's a soft tissue, and you have a contraction after the bolus passes. That's not included here. So you study it when it passes. You can do the things that you do, keeping the breath with the valve. You can flip down the epiglottis that covers the entrance to the airways. You can close the upper esophageal sphincter, which is the bottom valve that when you have the esophagus. So you can simulate basically real swallowing or not functioning swallowing. And you can test different fluids to see how they behave. And you measure the pressure at different points, and then you can also measure the velocity profile. And for doing that, this goes back to an earlier project that we had. That it actually started as a master project, which became a PhD project, that became research projects, and finally a company. And this is the incipientus that you have on the exhibition here. But that took like 20 years or so it's quite a slow process. But anyway, the idea is that you send in ultrasound pulses, and the pulses bounce on the fluid elements. Then you know where they bounced, and if you measure the reflected frequency, you also have a Doppler shift, and you know the speed. So you can get the complete velocity profile in about 100 milliseconds. And you can also run it through a tube. You can measure it on yourself, and you can measure in our throat model, so you get the velocity profile. And you can also calibrate and get the particle concentration. But anyway, if we go to some more swallowing in fluids, if you want to eat something as well, and solid foods are not very good for if you don't swallow okay, because they get stuck, you have PCs, people have problem chewing and so on. Unfortunately, this is much of what you have to look forward to, sorry for the gloominess again, in big parts of the world. You just mash all of your meal and you get it as a porridge, because that is easier to swallow. But it doesn't have to be like that. You can have texture-adjusted foods like this instead. So you have to adjust the texture so it's easier to swallow and the bolus keeps together. And this is called different things in different countries. In Sweden and most of Northern Europe, you have the different scales. There is also international scales. But the absolutely most common thing is timbal, and that's what you see on the pictures. You can see that they are soft. You can easily crush them. They are moist. It actually, you take the real food, like the green broccoli here, and then you mash it, homogenize it, you add eggs and starch, and then you heat it, so it becomes a gel-like, omelette-like structure, which is, you have the original taste, but it's reconstituted and have another texture. But then, when you do this, you don't really know if you modify the food, you don't modify the bolus. Probably it connects, because this is done through the years and people have perfected it more and more, but we wanted to test if can you really also have these good properties on the bolus that you swallow. So we did a study on this, that we asked people to chew different foods, real food, the timbal and gels, and then we asked them to chew until they wanted to swallow, but then spit out instead. And then we measured the properties of this bolus, and we also measured like the saliva you need and the amount of chewing you need, and we could see that, yeah, it does work, which is lucky, otherwise, yeah. So you need less saliva, you need to chew less. If you have the more and more you modify the food, gel is of course easiest. But also what was not that obvious, if you put different components together, like if you have a sandwich instead of eating just a cracker, it's much easier to swallow. And also there was this, we measured on an old group and a young group. It was like average age 35 to 70 something. And we expected that the older group would have more problems, but they were actually quite fit. So even if they have the things that comes with age, like less saliva, they compensated. So they have perfectly safe swallowing, even if they were older. So this is a good thing, right? You can go get older but still be fit and not have swallowing problems. So I have to quote Oscar Wilde because he really expressed this well. He said that age is a matter of the mind, and if you don't mind, it doesn't matter. Okay, so we also want to know what happens in the mouth, because, I mean, if you imagine you're a food company and you can sort of know everything what happens in the mouth, a flavor release, texture, and everything, you can really design the perfect food. And also from a scientific point of view, it would be fantastic. But as soon as you insert some sort of sensor in the mouth, you disturb the process, and you don't really know what is going on because it's another process. So we realized that we had to have something which is non-invasive, and fortunately, this additive E172 is iron oxide. You can get that magnetic. And if you have magnetic small particles, you can measure the rotation. So if you measure the Brownian rotation and the relaxation of that, you can actually connect that to the surrounding rheological properties, which means that you can follow, like microurology, just this is rotation instead of translation. So then we tested with the normal standard fluids, and it connects pretty well. I mean, you measure really linear viscoelasticity close to the small particle, and you do that also in aerometry, and it works fine. The setup is on the top here, where you have magnetic coils, and you have a small sample in between. But this is not very suitable for swallowing or for chewing. You can see this tau b is the Brownian relaxation time, which is in the simplest model connected directly to the Newtonian viscosity. There are other models which are more advanced and also includes elasticity. And then you can see the upscaled coils here, and you can see one healthy subject, as it says in the papers, who is going to try this. So I was chewing timbals and I was chewing gels with added iron oxide. And this is just initial results. But you can see that if you follow the gels, these also have two in-phase and out-of-phase components. So for the gel, you can see that there is an increase when you start chewing, then you have a decrease of the signal when the food is moving too far away from the coils. For the timbal, it's sort of hidden in the scatter of the signal, so you can't really say if it's true or not. So I mean, it's proof of concept. It works, but you need a better signal, and you want to cover the whole mouth. Now we can cover maybe 2-3 centimeters. Okay, so now let's switch into gels instead. I did gels as a PhD student, but I'll focus on one later application. I want to stick to Save the World theme or Save Somebody theme. Cartilage is a gel. And cartilage is what you have at the end of your bones that reduces friction and dampens the movements. It consists basically of collagen network, which is expanded with protein. So you can see the structure here, or part of it. So what you can do is, if you get an injury, it's difficult to repair. And if you get a degradation with time, which is commonly like arthrosis, which is common, you can't really do very much about it. You can repair smaller injuries, but not really larger ones. So the idea is that you take the cells that produce this extracellular matrix, you grow them on some sort of construct or mesh, and then you transplant them back to the body, because then you have your own tissue. There's no problems with the rejection of the tissue. You need the mesh because the cells has to adhere to something, otherwise they express the wrong type, so you don't get collagen, you get more like scar tissue if you just have them on the surface. You have to have some sort of 3D structure. In this case, it was polyglycolic acid fibers that degrades over a couple of weeks. So you can see that the fibers are covered with chondrocytes. They have produced collagen. They have also migrated out in the extracellular matrix. This is a couple of weeks old. So it works well. But then you want to see how strong is it with time? How long time do you have to have it in tissue culture? But that's not so easy to measure because it's a slippery substance and you have to put it in the instrument in some way. You have to use parallel plates, but you have to know how much is in contact with the plates because it's not, of course, not perfect cylinder. So then you have to build something like this, so you can observe how much of the upper part is in contact with the plate. And then you can get linear viscoelasticity. You can see that they form over time, and also the modulus goes pretty much linearly over time. This is tissue culture for 7 weeks. If you compare that to natural collagen, it's fairly close. So the composition is close, but the modulus is much lower. So it's not perfectly similar to the normal cartilage that you have in the body. But there is some hope as well. There is a surgeon close to Gothenburg who has done similar things, like hundreds of operations. But he takes a cell, multiply them, put them back, and put a piece of thin tissue over. So it's not a construct. And then actually the cells start expressing the right type when they are in the right environment. And you can actually heal this trauma or small missing piece in the body. So it should work. One problem if you want to have something to do or a new project is how to design this construct so you can actually have a much higher diffusion of nutrients into the tissue, because that is limiting the size. This was just 2 millimeters thick. If you go to 4 millimeters or 5, the center doesn't have the same structure as you want it to have. So if you can design a construct with tubes or something, that would be nice. That degrades over a couple of weeks. Okay, last part about solids. I mean, it's almost solids. I'm talking about melts. It's also following up from yesterday because this is protein melts. And the reason this other sort of society's challenge is that we eat too much meat because it contributes to global warming and it's not good for health. That's the short story. I mean, if you look more into sustainability, foods is about 25% of all the greenhouse gas emissions, and meat production is like 14.5%, and that's like the transport sector. So it's a lot. And also, we use a lot of land for production. So 70% here in Western Europe is used for feed for the cows. And if we would eat the food directly, it would be enough to feed the expected 12 billion people. And health effects, we eat, like this, Europe's almost 70 kilos per year. The goal is to decrease it to about half. If you look at the countries on the right, you can see it's a big spread. We shouldn't stop eating meat because meat is quite good as well. And everybody shouldn't eat the same lower amount because middle-aged men like me, we should eat less for sure. Women in reproductive age shouldn't eat that much less because it's also good iron source. And if you look at the countries at the bottom, they should eat more meat if they could have it. It's not black and white. And the plant-based alternatives are not perfect either because the iron bioavailability is pretty much zero. So we have to change that too. So another way of doing this is that you can grow tissue in culture, so you can grow it in the lab instead. It always gets much attention in media. There are some products, I think it's Singapore. Singapore has a good legislation for this. This is an example when we tried it. Roland was extruding fibers, and we extruded fibers from plant protein because our idea was that we could have the majority of the mass from plant proteins, and then we put muscle cells like icing on the cake to get it really meat-like. And it worked. It worked like the cartilage I showed you before. You can see that you have muscle cells that have migrated out. They have formed muscle tissue. But muscle tissue is not really meat. I mean, meat is more. It's connective tissue, it's fats, it's structure on several layers. But you can grow muscle cells, so that's okay. It's just very difficult to scale up because it has to be totally pure. I mean, no bacteria whatsoever, because this is perfect for growing bacteria as well. So big scale production is quite difficult, but not impossible. So it will come. So this is what we heard about yesterday, both from Sandra and some other talks. A lot of people are working with plant proteins on different aspects. And The great thing is that you can extrude them and you can form something that has a fiber structure similar to meat, similar to chicken meat. And you can do that mainly with legumes. You can also use gluten. I said that there are some concerns. We can solve that. But in addition to a normal extruder, you also need a cooling die, because it's in the cooling die the magic happens. That's where the fibers are formed. And I say magic because people know how to do this, but not exactly why they are formed. And there are different models, of course. It's a very active scientific discussion, so it's interesting to go to these meetings. You have to discuss a lot. The first one wasn't very successful. The other one, you can have physical interlocking of crystallites. I'll show you a picture of that. The third one I talk more about, because that's a combination of heat transfer and phase separation. The fourth one is new. That's the Swiss school that say that it's not phase separation, it's microcracks. And it's very difficult to show exactly what it is, because you want to look inside the extruder, but that is 150 degrees and 10 bars. So it's difficult to observe what's actually going on. Actually, I'm going to skip the rheological part here. That is a separate talk. It's quite difficult. If you have seen the poster by Felix, you see that it's really difficult to measure this. We also use closed cavity and normal rheometry, but food evaporates and then you have a different material. If you're very careful, you can get some good data, as Sandra showed yesterday. But this other model, which I'm not going to talk more than this about, this is interlocking of crystallites. And this is actually PTFE or Teflon. So I think this is quite hopeful. You can get fibers also in a plastic, which means that you should be able to use other proteins than just legumes. You just have to get the flow, the fluid dynamics right. But in the other model with the phase separation, I will show you some simulations, because then you simulate phase separation, Cone-Hilliard type, with this spinol decomposition, so you get the protein-rich phase and the protein-poor phase. And you have to assume the rheology. We use power law or LPTT if you want to also include the elasticity. We use COMSOL multiphysics, which is finite element-based, and then you get something like this. So this is the cooling die cross-section, and the y-axis is scaled, is 5 times bigger than it should be. And the first thing you observe is that you get the funnel, really, because you have cooling from the outside, then you increase the viscosity a lot, and that means that you have much warmer in the center. That also means you have lower viscosity and faster flow. So if you don't get the balance of cooling right, you have warm material just shooting through, like you have a core slip in the center. So you have to balance how much you cool. You end up in very long cooling dies when you do this. But you can see here a little what I talked about. In the center, well, you have a blue phase and a red phase, which are the protein-rich, protein-poor phases. In the center, you are over the critical phase separation temperature, so you just have a flow going straight through. Eventually, it will also be fibrous in the center. And it's quite coarse, but that is just a numerical issue. If you spend more computer hours, you can get also the finer structure. I should first talk about what you actually see when you get something out. It is difficult or almost impossible to measure inside the cooling die, but you can put some black dye and then you can see what comes out. And there you can see that you have actually some plug at the end, so it doesn't really shoot through all the way. And when it comes out, you can see that you have traces of the velocity profiles. Here, it's easy to say that, you have phase separation, you can see the different phases, but you can't really, because you have always put a bit of attention when you prepare the samples. So the phase separation with heat transfer, we think that actually works well. But we need something more, and presently we try to induce also slip or partial slip, because that would explain why you eventually get the flow and a different heat transfer along the cooling die. This is also one of these ultrasonic transducers that we have used to measure. I can say that it's possible to get the velocity profile inside the cooling die. It's difficult, but you can. But we wanted to compare the velocity profile in reality to the one we simulate to be able to validate the simulations. But it turns out that you don't have enough resolution close to the wall, so that's a tricky part. So I think I'm approaching the end of time, so I'll get you some conclusions. I promised you the secret to healthy aging. And you've seen some of them, the food is there. But I mean, you shouldn't smoke, that's obvious. I think you know this already. I'm going to show 4 of them. Probably you're very much aware, but like me, you don't always follow it. So you should eat according to science, and that's like more than 500 grams of vegetable and fruit today. How many does that? Not so many, maybe. I don't usually. You should also exercise quite a lot, and this is also the science collected. So it's 2.5 to 5 hours cardio. I mean, not a normal walk, something where you are breathing heavily. And you need to go to the gym. If you're over like 65, you should also practice your balance, because that goes down with age. And you should drink very, very moderately. Somebody has estimated all this is 5 years on this, but I think it's more important that for aging that you have a quality when you get older, so you can still do what you want to do. And of course, we should eat less meat. I don't think we should stop eating meat, but if we eat half the meat we eat today, that's what I got when I was a teenager, and I didn't really feel deprived of meat. That's what they have in much of the other parts of the world, so it's not so drastic. We can do this. And then, if you want my YouTube channel, it's here. Anyway, and also for young side, I wanted to say something, and these are my views, so don't only believe me. But I think it's important that you find what you like, because you are going to work with it all of your life. I mean, if gel structure neurology wasn't my life goal, but the PhD was pretty okay then. But after that, you have a possibility to choose. So think about that. And neurology, there is more than neurology, actually, even if you don't believe it. And I think you should treasure these other skills. Like if you like to travel, that's excellent. If you like other cultures, that will help you a lot. I mean, when you interact with other cultures, if you have been there and you know the customs, it would help you so much in the corporations. So take care about that one as well. And also, I mean, you have to discuss, but diplomacy is pretty good. So it's not good to start with that you're wrong. I heard that here as well. So discussions are good, but listen to the other person as well. Diplomacy is pretty nice. And why did I spend 30 years? Well, because it was fun. I mean, it's really nice people, and it's I really enjoyed all the time. But then also, I think it's good with mentors. Some people can do a career without them, but they are really good. And I want to mention 2. First, Anne-Marie Hermansen, who is a known name in food science. She helped me a lot, and I mean, she believed in me when I didn't believe in myself, really. So it's very good to have mentors who can give you advice, because you don't know everything. I published a lot together with Anne-Marie. I haven't published with Ken Walters, but he still introduced me to European neurology, which is very useful when you want to cooperate and you want to network and so on. Otherwise, I would never been the president of the European Society. So I'm very grateful that unfortunately Ken has passed away, but Anne-Marie is very much active, so you may see lectures from her as well. And then I thought everybody has pictures of the one they are collaborating with. I started, but then I thought, well, I do something, and this is a lot, but this is co-authors of my papers, and it's not the neurology board, and it's not a lot of you people, but I wanted to have one of these slides anyway. So I'm very thankful for all the cooperation, and I'm very thankful for this opportunity, and most of all, I'm thankful to you for coming and listening to me. So thanks.